this is going to be a story about my journey to becoming a research data steward at the Centre for Advanced Research Computing at UCL. So I call myself a microbiologist, and I've turned to being a data steward. So I'm going to talk about kind of how I got to become a microbiologist. So I did a little bit of a different first degree. So I loved horses and I loved science, and 18-year-old me just thought this was a great idea and nobody could convince me otherwise. Should have just done microbiology or biology straight, but I enjoyed the degree. And I thought I'd go on and apply for graduate veterinary medicine. While I was doing the degree, I didn't get in, and it was devastating at the time, but in hindsight, I'm glad, because I think I would have just gone into research anyway and had a lot more debt. So I'm glad I didn't get into veterinary medicine. And this degree actually did get me into microbiology, because I did a lot of veterinary microbiology modules. My undergraduate dissertation was on the healing of equine wounds, and that involved a lot of lab work with microbiology. So I went on, and also I learned horses are dangerous. I had a lot of nasty accidents while working with them and decided I didn't want a career working with these massive animals. So I went and worked with teeny tiny microbes instead. So I did a master's in medical microbiology at Cardiff Met, and here I got my first research publication, and this really got me thinking, oh, maybe I should go into a research career. So I went on and worked at a company called BBI Solutions as a conjugation and quality control scientist. So here we created the point of care tests that we've all come to know and love from COVID. So we created that red line where we conjugated antibodies to gold nanoparticles. And I also got involved in the quality assurance and quality control. So it kind of got me thinking about documenting everything. And industry is really strict on kind of how everything gets produced. So all the batches are the same and we know where everything's come from. So I then went on and did a PhD in bacterial genomics at Cardiff University. This was sponsored by Unilever. So we were looking at how the bacteria Pseudomonas aeruginosa is able to contaminate shampoo and laundry detergents, so common household products. And we were looking at how it becomes resistant and what antimicrobial mechanisms it uses. I also went to the University of Virginia and did a placement there, which was really cool. So we had a few publications out of this work. And what this really got me working with big data and managing large genomic data sets. So we wanted to create a reference genome for an industrial Pseudomonas aeruginosa isolate, so one that had been isolated from a Unilever supplied product. And we chose an isolate that they used in their testing of preservatives. So we created a reference genome, so this had to be really well annotated. And all the processes we used to sequence, assemble, annotate the genomes had to be really stringent for research. So this kind of got me working with large genomic data sets. I then went on and did a postdoc at the University of Liverpool. And I also did a placement in Memphis in St. Jude's Children's Research Hospital. So I've done a lot of collaborations, which has really helped kind of with the research and getting experience with that. And here I really honed in on my passion for data visualization. So I did experimental evolution models, and in order to kind of look at how bacteria evolve over time, we use these Muller plots. They produce nice, pretty graphs. They're really useful for visualizing your data and understanding large, complex data sets. So I made them into art, and it's won a competition to be on the front cover of this journal for 2025, so that was really cool. So we published quite a few papers during my postdoc. And again, this got me into working with large data sets again. So we did experimental evolution work. So these experiments ran for 2 years. So we collected a lot of isolates. So we were trying to evolve the bacteria Streptococcus pneumoniae that can live happily in your nose and not cause you any problems, but can then go into your lungs and your blood and cause disease. So we wanted to see how the bacteria adapts to different environments. And we used experimental evolution models with mouse models. So this kind of I really had to learn how to manage the project, plan the experiments, manage all the isolates, manage the data, manage the sequences. So it kind of got me really working with large data sets from the beginning right through to the end. And the project, so we published on this, and we had large data sets that we had to visualize and understand what was going on. And we wanted to identify specific genes that were really important for the bacteria to grow within certain environments. So these genes here were really important for the bacteria to grow and survive within the nose. And these being able to take the large data sets, visualize them, and identify the different genotypes using these Muller plots was really helpful. After that, decided I didn't want to stay in academia just for the short-term contracts and not knowing kind of what my job was going to be in the next year. So I decided to try and get permanent jobs. Went back to Unilever and worked as an R&D microbiologist. Then switched and went to the charity sector and worked for the Healthcare Infection Society, managing their research grants. That was really useful because I learned how to obviously manage the review processes and again, kind of learn like systems for reviewing grants and managing committees. And then I became a research data steward. So I wanted to kind of go back to a research-related role, but I wanted it to be permanent, and I didn't think these sorts of jobs existed. But I found this job. I remember seeing the title, Research Data Steward, and thinking, I don't think I'm a data steward. But then when I was looking at the projects, at the description, it kind of listed all the things like data management, working with the data lifecycle. And what I really liked is that they want you to sort of develop your data stewardship skills within your profession. So I applied and I became a data steward in March of last year, and I'm really enjoying it. And I think some of the key experiences, so I just want to summarize some of the key experiences that I've had throughout my career to kind of build my research data skills. I think working with genomic data and metabolomic NMR, large multi-omics data sets has been really useful. Managing experimental evolution data sets and visualizing complex data sets. Going through the publication process and the grant writing process has been really useful. And I think working in industry, in the quality assurance and quality control processes has been like really key. Working in bioinformatics and helping to develop a platform for data analysis. Mouse model work has been really important because you have to plan so much in order to use the minimum number of mice in order to get the best results. And then documenting methodology, handing over research projects to other students is really important. And then uploading data sets and metadata to domain-specific repositories throughout my academic career has been really important. So just quickly, some of the projects that I'm involved in at UCL. So we're currently working to migrate research data from cloud platforms to centralized UCL research data storage service. So we want to bring all research data together into the same place. We've developed a standardized way of doing this and to make sure that the research data is copied over and everything is safe for the researchers. And then I've also been involved in building and developing and delivering workshops. So we've got one, building data skills to tackle antimicrobial resistance. So we give an introduction to R databases and reproducibility for AMR epidemiologists. And then I've also worked in various other projects looking at common data models, such as OMOP, and trying to map that to different programs in order to be able to use OMOP to look at antimicrobial resistance and developing standardized bioinformatic pipelines for microbial genomics and data analysis. This is something that I'm currently working on to develop.